Okay. Are we ready, Violeta, to start? Is it?
Yeah, I think so. Now it is transmitting YouTube. So whenever you're ready, I think it's maybe good to wait another couple of minutes.
Okay.
Usually people start arriving a little late.
Okay, yeah. Thanks. Well, to give a couple of more minutes, I would like to just to introduce like the institute to you, Fabio. We are the Institute of Cell Physiology. We are based at the main campus of the National University, and we have PIs or researchers that study a wide range of fields in the biomedical sciences. It's mainly a basic science institute, and but we have like a whole neuroscience division and the basic science division where we have microbiologists, structural biology, immunology, I don't know, like developmental biology, and so help me if I am missing something. And in neuroscience, we have like at the molecular level, cognitive neuroscience and the like. So it is a wide audience.
I will try to be as clear as possible, but it's nice you have so many different expertise on campus, so that's great.
Yeah, so and this is our institutional seminar, so we all the departments come together to listen to science every Friday. So I think we are ready to start. So thank you again for accepting our invitation. Thank you everybody to join us, and thank you to the people also that is listening this seminar by YouTube. Please write your questions or comments, and we will read them. Only the people that is connected through this Zoom link can open the microphone and speak, but the people from YouTube, please communicate your questions, and we will we'll read them, and Dr. De Monti will answer them. Dr. De Monti is is an associate member, St. Jude faculty. That it's equivalent to a tenure track associate professors like to us. He studied the the bachelor degree at the University of Milan at the medical school in Italy. Then he did a PhD in Germany at the Max Institute in Dresden in cell biology and genetics. Then he did a postdoctoral stance in physiology and aging at Harvard Medical School. And he has done several very interesting contributions in the field recently of aging. We got interested in his work by reading his work in our lab meetings and also in the course that we gave about aging. And he has several important contributions in the protein quality control that protects signal from the skeletal muscle that protects from aging, and he has published his work in journals like Cell, Nature Communications, Cell Reports. He has also contributed to the understanding the influence of skeletal muscle on brain proteostasis and aging via myokines, and also the myokine signaling in nutrient sensing. And so that is like the main field in which he has contributed, and he's gonna share with us. He has received several awards. For example, the Hartwell Individual Biomedical Research Award, the Glenn Award for Research in Biological Mechanisms of Aging, the Allison Medical Foundation New Scholar in Aging Award, and several more. So please, Dr. Dimontis, I leave you with this. I send you the microphone. So thank you.
Thank you so much for the introduction and for the invitation. It's a pleasure to present my research today, and so today I will mostly speak about muscle to brain signaling and the work that we're doing on this in my lab. My lab also works on sarcopenias, on skeletal muscle aging. So I will briefly touch on the work that we do on sarcopenia in the first part of the talk, and then most of my talk will be on myokines and so how the muscle can communicate with the brain and with other tissues. And so, as I was saying, my lab works on these two different aspects, and so sarcopenia is the loss of muscle mass and strength that happens with aging. But apart aging, there is a loss of muscle mass and strength that happens in a number of other diseases, and it's really a huge list of diseases. So one of them is cancer. So there is the so-called cancer cachexia, where there is wasting of the skeletal muscle as well as other tissues like the adipose tissue. But also infections, neuromuscular disorders, and diabetes, they all lead to muscle wasting. And so that's not only a complication of these diseases, but is also an important determinant of survival. For example, in the cancer cachexia field, it was shown that if you prevent the wasting of the skeletal muscle without affecting the growth of the tumor, you can actually improve the survival of the mice that have these tumors. So again, improving the health of the muscle is improving the survival of the organism in the case of the cancer cachexia studies. But there are epidemiological studies in humans showing that the same is true in pretty much all the other conditions where muscle wasting happens. So if you can preserve the health of the muscle, you have beneficial effects on aging, for example. So the people with better muscle function, they also have a lower risk of many other age-related diseases. And so by studying sarcopenia and how the muscle in terms affects the whole body, my lab wants to understand more on how skeletal muscle health can have such a widespread impact on organismal health. And so this is how a sarcopenic muscle looks like. So you can have, you can appreciate that the size, the cross-sections of the muscle cells, also called myofibers, is reduced compared to young age. And there are a number of other deficits in the function of this muscle. So there are metabolic defects in mitochondria. So far, most of the studies were actually done on muscle wasting conditions that happen in young age. And what has been known from those studies is that the size of muscle cells, also known as myofibers, relies on the balance between protein degradation and protein synthesis. So typically in wasting, you have in muscle wasting, you have increased protein degradation and reduced protein synthesis. And so this is the reason why you get the small muscle cells that I showed in the previous slide. And so for the cure or the possible intervention that is proposed for all types of muscle wasting is that you should find a way to increase protein synthesis and reduce protein degradation, with the idea of increasing the size of the muscle cells and in turn inducing muscle hypertrophy. However, this is paradoxical if you think of this in the context of aging. And so there are many reasons to say this. So one reason is that we know that many pathways that promote protein synthesis, like the mTOR pathway, they actually accelerate aging rather than protecting from it. And we also know that, and so the proposal of increasing protein synthesis in that case will be quite difficult to understand how it would work against an age-related disease like sarcopenia, so the muscle wasting that occurs with aging. Likewise, if the proposal is to inhibit protein degradation, it's hard to understand how this could work in aging because the loss of proteostasis, so the accumulation of misfolded proteins, is a hallmark of aging. And so if you inhibit protein degradation, then you will prevent the degradation of those misfolded proteins, as well as the dysfunctional organelles that are a typical feature of aging and that you actually want to contrast. And so this proposed cure for muscle wasting is quite at odds with many of the things that we know about aging. And so either sarcopenia is a unique age-related disease for which all these hallmarks and all that we know about aging does not apply, or the cure that has been proposed for muscle wasting is actually not a good cure for sarcopenia, so for the muscle wasting that happens with aging. And so a core part of my lab is then investigating the role of protein quality control and whether it's important for sarcopenia. So whether what will be a good intervention for preserving the function of skeletal muscle with aging. And so to address these questions, I use the fruit fly Drosophila melanogaster. And there are many advantages to use this model organism. In the context of aging, a key one is the short lifespan, which is around 3 months. The limited genetic redundancy, so you have typically one gene rather than multiple paralogs, as it can happen in humans or mice. And we also have a fairly good conservation of the genome. So we have around 60% genes, genome, a level of 60% genomology comparing Drosophila versus humans. And as you know, we have also many genetic tools, so we can do RNAi and overexpression of any gene of interest, and we can do that tissue specifically. So we can do these interventions in skeletal muscle. We can also test drugs and environmental factors. We can study muscle with basically all the techniques that you can also use in other model organisms like mice. And I will show later on that we can also study intertissue signaling via myokines. And so to give again an introduction on Drosophila, so there are several stages of the Drosophila life cycle at which you can study muscle function and features of muscle biology. So in the embryo, you have the initial phases of muscle differentiation with the muscle precursors or myocytes diffuse, myoblasts diffuse. And then you have the larval stages where you have a massive growth of the muscle cells. So you see these are tiny muscle cells that form at the beginning of development. And over the 5 days of larval development, you have a massive growth and you end up with these big muscles at the end of larval development. So you see these are actually 2 single myofibers. So one is here and the other one is there. These are 2 stereotypical myofibers that we analyze for our studies. In the pupa, there is extensive muscle remodeling, and eventually there is a new set of muscles that is made for the adult flies. And so in the adult, so typically we study interventions that regulate the muscle growth in the larva, and we use instead the adult flies to study interventions that regulate muscle function and the maintenance of those muscles over the lifespan, and how those muscles or muscle-initiated signaling in turn affects other tissues. And so to test the initial hypothesis that I introduced earlier on, so whether a protein degradation is indeed important for muscle wasting in flies as it was proposed in humans, what I did was to do a very simple experiment. So I modulated the levels of a transcription factor called FOXO, which is really a canonical transcription factor that acts downstream of insulin signaling. So it's inhibited by insulin signaling and activated by lack of insulin or by starvation and stress. And it's a known inducer of muscle wasting in mice. So if you activate FOXO in mice, you induce myofiber atrophy. And what I found is that the same is true in flies. So when you activate FOXO, in this case genetically by expressing a FOXO transgene, you reduce the size of these two stereotypical myofibers that I introduced in the previous slide compared to a control intervention. And so on this basis, I was thinking, okay, what's known in mice is happening exactly in the same way in flies. And so muscle wasting indeed happens when you promote protein degradation. And FOXO promote protein degradation by activating autophagy, as well as by improving the function of the proteasome. So then I wanted to know what happens if you activate FOXO in the adult muscle. And here came the surprise. So I was expecting the FOXO activation would lead to muscle wasting, to poor survival of the flies, and I found exactly the opposite. So with FOXO activation, I found that the flies were longer lived, they were having an improvement in muscle function. And when I look at these muscles, I found that they were also having a better protein quality control. Specifically, what I found is that in mid-aged flies, so at 5 weeks, there was an accumulation of protein aggregates in control flies. And so you see these speckles there, they're aggregates of polyubiquitinated proteins. They're also positive for a marker called the P62, which is an autophagic adaptor protein. And so these are basically, this is a phenotype that is well known to happen in the brain and with also pathogenic proteins in the brain, but was unknown to happen in the muscle. And so these really suggested me that the muscle age in a very similar manner like the brain, and that therefore all those aging hallmarks that I introduced earlier on are likely to be applicable to the skeletal muscle as well. And in fact, with activation of FOXO, which promotes protein degradation, there was a removal of the aggregates that you find in the control. And I did also many other experiments that indicate that FOXO activates autophagy, and the activation of autophagy in this case is not detrimental as originally anticipated. So it's not inducing muscle wasting and reducing the function of the muscle, but I'm sorry, is there a question? Okay.
No, sorry, someone had opened it.
All right, sorry. So in this case, the activation of autophagy is not detrimental as originally anticipated for the studies on muscle wasting in mice, but is actually beneficial because you remove these misfolded and aggregation and aggregated proteins that you see in the control. So you improve the protein quality of this muscle, and this in turn improves the capacity for force production. And surprisingly, there was also an effect on lifespan. So by improving the health of the muscle, we were able to extend the lifespan of the fly, even if the activation of FOXO was restricted, was specific only for skeletal muscle and was not happening in other tissues. And so based on these studies and other studies that I don't have time to present today, the intervention that we think is most likely to work for sarcopenia, and by the way, this conclusion is also based on studies we did in mice, so it's not fly specific. So the intervention that we think is ideal is an intervention that promotes the degradation of misfolded protein in skeletal muscle, so that you can again preserve the functionality of the contractile apparatus as well as of the muscle proteome, and in turn promote muscle force production. And so basically, to look at this simplistically, we would like to propose that muscle aging should be treated like brain aging, and so it should be treated like a neurodegenerative disease. And like those conditions, we propose that removing misfolded proteins and preserving the functionality of the proteome is really key for preserving skeletal muscle function with aging. So I mentioned that this conclusion is not based only on FOXO and not only on flies. We did many studies, and I just want to show one slide on one of these studies. So to look at other interventions that regulate muscle cell size, we did a screen in flies, which is again something for which Drosophila is well known for. In this case, we screened 646 RNAi lines targeting all the ubiquitin ligases, which are key regulators of proteostasis by ensuring the normal turnover of proteins as well as by degrading misfolded proteins. Out of the screen, we found novel regulators of myofiber size. So the RNAi for these E3 ligases was inducing myofiber hypertrophy. There were also RNAi for E3s that were inducing atrophy. But what was remarkable is that when we tested these E3 ligase, these interventions that induce hypertrophy, when we tested them in the adult flies, we found that all of them, they shorten lifespan and they reduce muscle function, and they do so by inhibiting protein quality control. So again, this slide is again showing that the conclusion I presented before is true not only in the context of FOXO and flies, but is also a general conclusion based on many other genetic interventions that we tested in my lab, in Drosophila as well as in mice, and is also supported by studies in by other groups, where they've shown that, for example, if you inhibit autophagy, you have accelerated aging and decreased muscle function. Again, contradicting this earlier hypothesis that to prevent muscle wasting, you'd have to inhibit autophagy and in general protein degradation. And so that's what I wanted to mention briefly about the work that we do on proteostasis and the importance of protein quality control in the muscle. Now, in the rest of my talk, I will speak on how the muscle, also in response to protein quality control, is able to interface with other tissues and regulate, in particular, the aging of the brain. And so the work on muscle-to-brain signaling that we do is broadly part of a big area of the aging field, which is on intertissue signaling. And there are many studies on this in flies, but also in mice and in C. elegans. And basically, the general idea is that for an organism to age well, you need proper communication between tissues. So if a tissue can communicate its needs or its stress status with other tissues, this leads to the initiation of systemic changes, behavioral, so behaviors or stress responses that benefit the whole organism and that improve the health span of the organism. So that's really a general theme that is quite well studied and in the aging field in many organisms. And one example of these studies is, I'm sure you're familiar with the studies that describe a quite striking effect of parabiosis on the aging and the function of many tissues, including the brain. And so in these studies can be done by transferring the blood from a young donor to an old recipient. And as a consequence of this plasma or blood transfer, there are many benefits that are observed in the brain, and you see some of them listed here. This experiment can also be done by adjoining the circulation of a young and an old mouse in a so-called parabiontic pair. And also in this case, the old mouse in the pair is showing signs of rejuvenation, including improved brain function, whereas conversely, the young mouse has experiences detrimental effects from this connection with the old mouse. These benefits are not due to the surgical procedure because you don't see any of those benefits in the isochronic pair, so young mouse, or in the isochronic pair with 2 old mice that are connected together. So this is to say there is clear evidence that circulating factors are beneficial for the brain. It's unclear where these circulating factors are coming from. And so what I would like to propose is that at least some of them are coming from skeletal muscle. And the reason to propose this idea is that we know the muscle contraction, so exercise, is one of the most potent interventions to delay aging and to preserve cognitive functions. And this happens via the induction of local effects in the muscle, as well as via changes that are induced in the brain. And so how this can happen, how can muscle exert such a function on the brain? So one possibility is via factors that are secreted by the muscle. These signaling factors are called myokines, and they can be cytokines and growth factors, but also metabolites, as well as many other types of unconventional signals like vesicles and lipids, for example. So again, many different types of molecules secreted by muscle can have these signaling functions. The signaling interactions of this muscle has been studied in fairly good detail with other tissues like the adipose tissue and to some extent also the pancreas and the liver. But I think what I'm primarily interested in is the interaction of the muscle with the brain, because again of this interconnection between exercise and cognitive function, and also because it's relatively underexplored compared to the other target tissues that I introduced earlier. Beyond aging, or in the context of aging, but even beyond, in the context of the normal physiological regulation of the function of the organism, we know that many tissues contribute to this intertissue signaling. Is not only the muscle, obviously, and the brain is considered a bit like the receiver center for many of these peripheral signals. So that's an additional reason for being interested in muscle to brain signaling, again, with the brain having such a central control that will be quite important to better dissect this interaction. And an example of the brain having a key control of systemic function is the regulation of feeding behavior. So many tissues, and in particular, we know a lot about the pancreas, the gut, and the adipose tissue. So many tissues, they can send the signals to the brain to indicate what is the status of their energy stores. For example, the adipose tissue can secrete a satiety signal called leptin that signals to the brain that there are sufficient energy stores in the adipose tissue, and so that there is no point to keep on feeding, and so that the brain can signal that the entire organism can stop looking for food and feeding. And so when looking at this very well-known paradigm in physiological regulation, my question is, what is the role of skeletal muscle? So like these other tissues, skeletal muscle has high energy demands, particularly during exercise, but also at resting levels. And so I find interesting that such an important tissue which is the most abundant tissue in the organism with a high energy demand is not in the picture. And so the other reason to believe that skeletal muscle may play a role in feeding behavior is that several myokines, so several of these signaling factors that are made by muscle, are already known to be regulated by nutritional cues, for example, in response to lipids or to nutrient sensing pathways like the mTOR pathway. And so the question is whether skeletal muscle can contribute to the central regulation of feeding behavior that is affected by the brain, via myokines or via signaling factors secreted by skeletal muscle. And so in this slide, I'm summarizing a study that we published, and the first author is actually she's actually from Mexico. She was a member of my lab that again did the study. And so in the study, we found that there is, in flies, we found that there is a myokine called DPP, which is homologous to BMP2 and BMP4 in humans. And we found that DPP levels increase in response to mTOR signaling, so in response to nutrient sensing. Conversely, if you inhibit, if you have nutrient scarcity, and so, or you inhibit mTOR signaling, you have a reduction in the expression of DPP. And what we found is that DPP is a key myokine for the regulation of feeding behavior. And this occurs by modulating DPP BMP receptor signaling in dopaminergic neurons. And we found, so if we inhibit specifically in skeletal muscle the production of DPP, we have a consequent increase in feeding behavior. So the nutrient scarcity in the muscle signals to the brain that it should be looking for food. And we found the same phenotype not only by inhibiting DPP receptor signaling, DPP production in the muscle, but also by reducing DPP receptor signaling specifically in a cluster of dopaminergic neurons. And this modulation of feeding behavior was due to the transcriptional modulation of tyrosine hydroxylase, which is the rate-limiting enzyme for the synthesis of dopamine, which is in turn regulating feeding initiation. So with this inhibition of DPP and DPP receptor signaling, the flies were spending more time nearby the food compared to areas of the tube that were farther away from the tube, and were accepting food when they were presented with food. So these are, again, classical signs of feeding initiation and foraging. And overall, then the study starts to address the question on how skeletal muscle regulates feeding behavior by signaling to the brain. The main area of my lab, though, is to understand how muscle to brain signaling can affect the brain in the context of aging, and especially if can be beneficial and delay brain aging, a bit like I showed you is done by exercise. And so I've shown you earlier on a slide where I was activating FOXO specifically in skeletal muscle. And I was showing you that there is an improvement in muscle function via an improvement in the protein quality control of the skeletal muscle compared to the control flies with no FOXO activation. And so these, the activation of FOXO was targeted specifically to skeletal muscle, and we have many genetic tools to do that in flies. So it was very surprising to find that the improvements in proteostasis were happening not only in skeletal muscle, but also in other tissues of those flies. And so we look at the retina, the brain, and the adipose tissue. And what we found is that there are protein aggregates in control flies that are when they are old. So you see here the retina, the brain, and the adipose tissue. Those speckles, they are aggregates of polyubiquitinated proteins. But when you were activating FOXO, specifically in skeletal muscle, you were preventing the accumulation of these protein aggregates. So in the FOXO flies, there were no or very few protein aggregates compared to the control flies in the retina, as well as in the brain and in the adipose tissue. And so this finding was indicating that the lifespan extension that is seen in these flies is also associated with an improvement in a protein quality control in a number of tissues. And again, my lab is particularly interested to understand how such signaling is happening to the brain and to the retina. And so I mentioned that exercise is already well known to improve cognitive functions. And there are many components of exercise. So there are many metabolic changes that happen with exercise, but one that is normally not well considered is the fact that there is a lot of mechanical stress in the muscle. So there is so muscle contraction by itself results in the misfolding of many proteins. And there are some that are particularly sensitive to such misfolding, such as phylamine and other components of the contractile apparatus. And as a consequence, it's known that exercise induces many stress responses in skeletal muscle. One of them is the unfolded protein response. And so the question that we ask in the lab is whether what's happening if we mimic this component of exercise? What's happening to the flies if we induce a moderate perturbation of proteostasis in skeletal muscle? And so for these studies, we then utilized transgenic RNAi targeting a core component of the proteasome, which is a key cellular system to preserve protein quality and to degrade misfolded proteins. And we did it again tissue specifically with one of the genetic tools that we have in flies to target these interventions to skeletal muscle. And what we found surprisingly is that if you drive the RNAi for pros-beta-1, so for this core component of the proteasome, we have very little changes in proteostasis. And so the way we are investigating proteostasis is to look at the pattern of polyubiquitinated proteins. So in young flies, you have so this smear that you see here is actually the pattern of proteins that are polyubiquitinated and that accumulate in insoluble fractions. So these are basically corresponding to the protein aggregates that I've shown earlier on in the immunostaining. And so what you can appreciate is that with increasing aging, you have a progressive increase in the accumulation of these insoluble polyubiquitinated proteins. Again, showing basically what I've shown earlier on in the confocal images, you have a progressive increase in the amount of those protein aggregates that contain polyubiquitinated proteins. And this other marker is called P62 in humans. It typically follows the polyubiquitinated protein. So it, again, is reinforcing the fact that there are protein aggregates that accumulate with aging. And so coming back to the intervention that we are doing, the pros-beta-1 RNAi, we see no changes in proteostasis. And what we found by RNA-seq is that there is a transcriptional stress response that is activating in the muscle by this mild decline in proteasome activity. And this stress response is based on the expression of proteases, which are already known from other studies to have the capacity to compensate for reduced function of the proteasome. And in addition, there is also the induction of chaperones that can help protein maintain their proper folding. And so in muscle, with this mild perturbation of proteasome function, there is not much happening. And we think that this is due to the activation of this local stress response. But then based on my earlier studies, the question is what's happening in other tissues, and in particularly, is there any effect on the retina and the brain? So is there any cell non-autonomous effect of this muscle-specific insult to the function of the proteasome? And so to address this question, we use the same assay that I introduced earlier on for the muscles. We look at the pattern of polyubiquitinated proteins, in this case from head extracts, which are enriched for brains and retinas. And so during normal aging, what you can see is that there is a gradual increase in the levels of polyubiquitinated proteins. So older brains from older flies, they have more insoluble polyubiquitinated proteins compared to the brains of younger flies. However, the brains of flies with muscle-specific proteasome stress, so pros-beta-1 RNAi, they have a lower levels of insoluble polyubiquitinated proteins compared to the controls. suggesting that induction of proteasome stress in skeletal muscle is improving proteostasis in the brain, and so is reducing the accumulation of these insoluble polyubiquitinated proteins. So we found this result with this driver, which MSC-GAL4, which is a constitutive driver, but we got the same result also with a drug-inducible driver. So in this case, we induce the expression of PROS-beta1 only when we add a drug called RU486 to the food, compared to flies that have where this transgene is not induced because there is no drug in the food. Also, in this case, you can appreciate there is very little accumulation of polyubiquitinated proteins compared to the isogenic control, whereas you don't see these improvements in flies that drive a control transgene called white RNAi. So in this case, you have similar levels of ubiquitinated proteins if you have the drug compared to the flies with no drug. And so the conclusion, again, that with multiple genetic systems, we were able to show that there is an improvement in the proteostasis of the brains and retinas in response to muscle-specific proteasome stress, either drug-induced, like in this case, or constitutive. And so we went on to do immunostaining, in this case of the retinas. Again, here you can see again the same protein aggregates introduced earlier on. These red speckles are polyubiquitinated proteins that are insoluble and that are sequestered away in these structures. And you can appreciate that when you induce PROS-beta1 specifically in skeletal muscle, you have very little accumulation of those polyubiquitinated protein aggregates compared to the same flies in which there is no drug added to the food, and so there is no expression of PROS-beta1 in the skeletal muscle. And so again, this is showing with a different assay what I introduced earlier on, that proteasome stress in skeletal muscle is preserving proteostasis in the brain, in this case in mid-aged flies. And I don't show it today, but we got the same result with a number of other transgenes. So we did RNAi for all these other canonical components of the proteasome, as well as for an associated pathway, which is called VCP. And we always got the same result. So there are reduced levels of proteasome substrates in the brain and retina. So we know that now we know that that in response to proteasome stress in skeletal muscle, there is an improvement of protein quality control in the retina and the brain. And so the question that we wanted to ask is whether this is dependent on stress-induced signaling factors made by the muscle, so myokines. And so to address this question, we did RNA sequencing of skeletal muscle with different RNA interventions, again, that knock down proteasome components, so PROS-beta5 or PROS-beta1 RNAi, as well as VCP, which is called TER94 in flies. And there were several secretive factors that you see listed here that were differentially regulated. Some of them consistently across these different interventions that induce this common protective response on the brain. And in yellow, I'm highlighting those for which we did some follow-up experimental validation. So we modulated their levels either via RNAi or by overexpression to see what was the consequent impact on brain proteostasis. And the key one, or one of the key ones that we found to have a role, is called AMIREL. So AMIREL was upregulated by stress. So you see here the fold regulation. That's the log to fold. So is quite high. Was quite highly induced by stress. And so then what we ask in this case is whether overexpression of AMIREL by itself in the muscle, so in the absence of any stress, is able to benefit the brain. And so basically we're asking whether mimicking the upregulation of amiral in skeletal muscle is having beneficial effects on brain proteostasis, like we have seen in response to the induction of proteasome stress in muscle. And what we found is that this is the case. So with the drug-induced expression of amiral, we have less polyubiquitinated proteins in the brain of old flies compared to the isogenic control that has the same genotype, but no transgenic expression because there is no drug added to the food. Whereas instead, the RU486 drug, which is again this drug that we use to induce transgenic expression, is not regulating proteostasis when it's driving no transgene, like in this case. We also got the same result in a context of with a background of Huntington disease. Also in this case, there were fewer polyubiquitinated proteins accumulating when amiral was induced in skeletal muscle compared to control flies where there was no amiral upregulation. And so the conclusion is that mimicking what stress does, so the upregulation of amiral in skeletal muscle, is able to preserve proteostasis in the central nervous system during aging. We went on to test this hypothesis via immunostaining of brains and retinas, and we found the same result. So when you have activation of amiral, specifically in skeletal muscle, you have fewer protein aggregates that accumulate with aging compared to control interventions where there is no amiral induction in the muscle. The same is true for the retina. Again, very few aggregates compared to the control interventions. And the same is true not only with the drug-inducible system, but also with a constitutive expression with this other driver called MSCGAL4. Very few protein aggregates accumulating compared to the control, both in the retina as well as in the brain. Again, that's the conditions where you have more amiral in the muscle and there are very few aggregates compared to the control. And so the conclusion is that the stress-induced myokine amiral is one of the signaling factors made by the muscle that mediate these responses. And so what are the mechanisms involved? Amiral is, so we went on to do RNA-seq of the brains, and we found that there are several protective genes that are induced in the brain by amiral. These include chaperones, such as the small HO protein crystalline alpha beta, which is called HOP23 in flies, as well as many proteases that you see in blue. And so I mentioned before, the proteases were also induced locally in the muscle, and there are known already from other studies to compensate, at least in part, for conditions where there is a decline in proteasome function. But how is amiral able to induce these protective responses? So amiral is an enzyme, so it's not a typical myokine like cytokines and growth factors, which is the majority of the myokines that have been studied so far. It's actually an enzyme that was known for its function in the gut, where normally degrades starch into disaccharides, and so into maltose. And so that's the canonical function of amiral in the gut, but in this case, we are looking at an enzyme that is made by skeletal muscle, so completely independently from the gut and in response to stress. And so the question is whether amiral, which is again an amylase, whether amiral acts in the same way as it was described in the gut. So is it able to produce maltose, and in turn, is there any role for maltose in proteostasis? And so the first thing that we did was to measure maltose levels, and we found that in response to proteasome stress in muscle, you have an increase in maltose levels in the flies, as well as, and you see that also when you induce amiral in skeletal muscle. I should also say there were quite a few papers already showing that maltose is typically induced in conditions of stress in flies and a number of conditions. including, for example, stress induced by A-beta peptides associated with Alzheimer, as well as starvation and many other stressful conditions. And so maltose levels are increasing upon conditions that induce amiral. So the initial conclusion is that amiral that is induced by stress in skeletal muscle seems to work in a similar way as the amiral that is known to digest starch in the gut. So how is maltose eventually able to play then a potential protective role? So there is a transporter for maltose, which is called SLC45. And this transporter is present in many cells in the organism, including in brain cells. And so what we ask is whether we wonder what is the effect of RNAi for this transporter called SLC45 in flies, as well as is the same name that it has in humans. And so what we found is that if we knock down SLC45, so the maltose transporter specifically in brain cells with this LAVIGAL4 driver, we find that we reduce the expression of key amiral-induced genes, such as this chaperone called SOP23, suggesting that the SLC45 maltose transporter is necessary for the transcriptional regulation of the protective genes that are induced by muscle secreted amiral. We also look at proteostasis, and if we do RNAi for SLC45 specifically in the brain, we see that there are more polyubiquitinated proteins compared to the controls, and you see the same by immunostaining. So again, the SLC45 RNAi has more polyubiquitin protein aggregates compared to a control intervention. The same is true also if you look at SLC45 null flies, again, more polyubiquitinated proteins compared to the control. And if you overexpress amiral in the skeletal muscle of SLC45 null flies, you don't have any benefit. So if you look at the polyubiquitinated proteins here, they are fairly similar to those that you get in the control. So amiral induction in an animal that is null for SLC45 is not beneficial. Again, indicating that you need a functional maltose transporter for amiral to be beneficial in aging. And so the conclusion is that muscle induction of amiral in response to stress increases the levels of maltose, and maltose via its maltose transporter of the SLC45 family is inducing the expression of protective chaperones and proteases in the brain and in the retina, and this in turn improves protein quality control. And then summarizing also studies that I'm not showing here. So we testing several of these chaperones and proteases by itself, and we found that they are indeed protective. And that this is true not only in the context of normal aging, but also in models of tauopathy and Huntington disease in flies. So then the final question that I would like to show today is whether there is any conserved role of maltose for proteostasis in human cells. So we found that if you treat HEC293 cells with maltose, maltose preserves proteostasis in cells that are choked. So normally you have very few polyubiquitinated proteins in HEC293 cells that are insoluble in normal conditions, but we get this massive increase when we choke the cells. But if these cells are pretreated with maltose, we have very little accumulation upon a choke. And these effects, they are not reproduced if we have glucose, which is a isoenergetic treatment, or an ACL, which is controlling for any effects of maltose on osmolarity. So again, these effects are not merely due to energy supply, which should be reproduced by glucose or by changes in osmolarity. Also, if you treat with another disaccharide, such as celobiose, you don't get those effects like you see here with maltose treatment. And lastly, if we knock down the maltose transporter with sRNAs for the two highly expressed member of the SLC45 family, we reduce the protection that you see with just the plain maltose treatment, suggesting that indeed maltose preserves proteostasis in each CHOC cells in a SLC45 dependent manner, also in human cells, like in this case. We then did a final test in human brain organoids that you see shown here. So these are cortical brain organoids, and they are a model for studying proteostasis and many diseases. Also, in this case, what we did was to compare organoids that are shocked, and you see that this results in a huge increase in the insoluble polyubiquitinated proteins compared to organoids that are not shocked, which you see here. But also in this case, we found that pre-treatment with maltose preserves proteostasis and impedes the accumulation of insoluble polyubiquitinated proteins compared to organoids that are not treated with maltose. And so the conclusion is that maltose preserves proteostasis also in each shocked human brain organoids. And this preservation of proteostasis also leads to a better preservation of spontaneous neuronal activity. So you see here in yellow and orange, the organoids that are treated with maltose, they have a better recovery from each shock. So you see neuronal activity goes basically down with each shock, but it recovers much faster in the organoids that were treated with maltose, again in yellow and orange, versus those that were not treated with maltose that you see in gray. And so the conclusion is that maltose preserves proteostasis, and this in turn results in a better preservation of neuronal activity as well. And so the conclusion is that in response to a moderate stress in skeletal muscle, which again is an important component of exercise, we have the induction of protective stress-induced myokines. One of them is the amylase Mirel, which increases the levels of maltose, which via SLC45 maltose transporter induces the expression of protective chaperones and proteases in brain and retinal cells. And this in turn improves proteostasis in these cells during aging. And so with this study showing how moderate stress induced in skeletal muscle can actually benefit the brain during aging. And with this, I would like again to thank you for the invitation to speak today, and to thank all the members of my lab, in particular in Mamta, Rai, that did the last part that I presented. Thank you so much.
Thank you so much for your very interesting talk. We are open to questions. Does anyone in the audience have a question? Yes, Lourdes.
Thank you very much for your very interesting talk. I would like to know if you checked whether the effect of maltose is mediated also by the expression of FOXO, or it has nothing to do with FOXO?
Yeah, that's a great question. We didn't really look in detail into that, but FOXO is also induced by stress, so it's a likely mediator of some of these stress response pathways. There was another study showing that a TGF-beta ligand called DODL is actually induced by FOXO, and that could be one of the mechanisms via which FOXO is preserving proteostasis. But you're right, FOXO may also play a role downstream of proteasome stress. The transcription factor we studied in this paper, and I didn't show it today, is CBP. So that's the transcription factor that we found to modulate amiral expression, but again, FOXO is well likely to be involved as well.
Okay, thank you.
Thank you. Gabriel?
Yeah. Hi, thanks. Very interesting, your seminar.
Thank you.
I'm curious to know if you have explored the other part of the balance that you show us. the synthesis of proteins and the effect of this on aging?
Yeah, so that's another great question. We didn't really work much on that, but it's an important point. So preserving protein synthesis is, again, one of the components for preventing sarcopenia, and they could have also systemic effects because it's also a component of that follows the response to exercise. The little bit of work that we did is on 4-EBP, which is normally considered an inhibitor of protein synthesis, but in reality has a differential effect on protein synthesis. So it's generally repressing protein synthesis, but is promoting the translation of some RNAs. And so that one is well known to delay aging. And actually, we got the same response that I showed for FOXO. We got the same response with the muscle-specific activation of 4-EBP. We also got preservation of brain proteostasis. And again, in that case, we don't have a specific factor mediating those effects, but is likely mediated by the preferential synthesis of some myokines. So you're right, protein synthesis, and more than the bulk protein synthesis, the differential synthesis of protective factors is likely to be part of this picture as well.
May I add a couple of more questions, if
Yes,
possible?
yes.
So the other aspect that is quite interesting to me, when you want to make a parallel between the protein aggregation observed in brain and what happens in muscles, would you expect that? I mean, I'm not an expert on that field, but I've read and participated with some groups that are expert on that field. Most of the efforts for preventing aggregation do not seem to be the key for treating neurodegeneration. Remember, for example, the antibody against the APP fragment, that didn't work out, among many other ones. So the question is, it seems that aggregation might not be the relevant part, but a secondary consequence of something that is not working well. Do you think that that is the same in the muscle, or here the story is different?
Yeah, so that's again another great point. So for the studies you were mentioning in Alzheimer's, it's true there were many antibodies against A beta peptides that initially failed. However, I think this year there was a new treatment that is actually showing some promise for Alzheimer's disease. And I think compared to the previous treatment, my understanding is that it's more effective in reducing A beta levels. So the idea is that those previous treatments, they failed because they were not reaching a sufficiently low enough level of A beta peptides. So I think there is an important role of proteostasis in neurodegeneration. We think is also very important for the muscle. At the same time, you're right that proteostasis is not the only thing. Based on the hallmarks of aging that I showed earlier on, those are likely to contribute to the aging of any tissue, including skeletal muscle. And, my talk was only on proteostasis, but metabolism will be very important, mitochondrial function, DNA damage. So in my talk, I just show an aspect, but you are right. There are many other components of muscle aging, and they are all interconnected. So DNA damage could lead to defects in proteostasis, but we think that proteostasis is not just a marker, and we think we can have a causal role in muscle aging. And I think the muscle is maybe the best example because it relies so much on the contractile apparatus and is so rich in proteins compared to other tissue. So maybe it's actually the best tissue where we can find striking examples. So now defects in proteostasis limit aging, but I agree there are many other components that go beyond the proteostasis.
Okay. My last question would be the induction of appetite. Is that associated with amino acid or protein intake, or is not? You haven't reached that level of definition on the appetite
for the studies on foraging. Yeah, so the flies, in that case, they have a mixture of different nutrients. So there are proteins, carbohydrates. We didn't do a test where we proposed different nutrients. That would have been interesting aspect to explore as well. So in this case, it's the standard food where there is everything. So in that case, we found an effect on feeding initiation, but you are right that we could have differential effects depending on whether the food is rich in carbohydrates versus proteins or versus lipids. So that's an important aspect which unfortunately we didn't explore so far.
Thank you very much.
Thank you. Fernando?
Thank you very much for this exciting talk. I have two questions. The first one is, shall we humans begin to look into the levels of maltose in our blood?
Yeah, so so Amiral is actually conserved in humans, and I don't do any studies in humans, unfortunately, but that's that would be very interesting to look at. So in humans, the homologue of Amiral are called AMI 1A, and I think there is a 1B as well. So what I know is that in mice, the levels of AMI 1A go up with aging. So it looks like it's normally induced not only by proteasome stress like we saw in flies, but also by the stresses that are associated with aging. And so we see it go up both at the mRNA and protein level. I think was maybe a threefold upregulation in the tibialis anterior muscle of skeletal muscle of mice. But we didn't do any other studies. It will be very interesting to see whether there is any modulation of the Amiral homologues in humans and mice with aging, and eventually whether it can be protective as a therapeutic myokine.
Yeah, it would be very interesting to see what happens to a mouse in which you continuously administer either the amylase or the maltose. Have you tried those experiments?
I want to try. I didn't have funding so far, but I'm hopeful I will be able to have the funding and the workforce to do that. But that's the goal, to examine whether the same pathway can be protective in mice, and so we could do the muscle-specific knockout of AMI1 to see whether if we present this age-related increase, whether again that's worsening aging. And then we can do the converse, we can overexpress it and see whether we protect, again, the brain with aging. And the SLC45 transporters are also interesting. There are SNPs associated with Huntington disease. So there are indications out there that hypomorphic mutants can worsen proteostasis like we've seen in flies. But again, unfortunately, we were not able to study this so far, but that would be a great goal.
Okay, one final question is, what happened to all the other myokines that you didn't talk about and that were also regulated in your system? Those that you made us, how did you call them in your slide?
Yeah, so there were several that were upregulated. So I guess we can call them stress-induced myokines. At some point, I was thinking to call them stress kines, but I think either way is fine. And so there are others that we know are also important. So there is another one we are working on, which was listed there, which is called CGA093, which is actually another enzyme. So again, another atypical myokine is actually a lipase. And we're looking at how it remodels the lipidome and how the modulated lipid species are actually able to have any protective effect. Because like for Amiral, we find that if we mimic the upregulation of these lipase, we can actually preserve brain proteostasis with aging. But again, it's a completely different molecule from Amiral. So it's not an amylase, it's a lipase. So we are looking more into the lipids that are modulated.
Thank you very much.
So you're right. Sorry.
Thank you very much.
Oh,
Yeah, that's
thank
fine.
you.
Because it seems like maltose and amyl cannot do the whole job. You need to have probably a concerted activity of all the myokines, and plus all the other cytokines that come from other organs.
Yeah, so it really looks like this is the case. There are multiple myokines that can be protective. I didn't speak of the opposite, which is also well possible, that in other conditions, for example, with inactivity, you could have detrimental myokines that are made by unhealthy muscle, and that those could actually worsen brain function and proteostasis. So that's also something interesting. So, there is a lot of emphasis on exercise and protective signal, but it will be also interesting to understand also how a sedentary lifestyle and an inactive or atrophic muscle, how they can actually be detrimental, not only by not producing protective myokines, but also by making myokines that are toxic or detrimental for the brain. So that's another aspect I didn't speak about today, but is also, so it also works in the opposite way as I presented today.
Thank you.
Thank you. Marcia?
Gracias, Susana. Thank you for your talk. It was very interesting for me also. Is this alpha amylase similar to the ones produced by pancreatic acinar cells?
I would assume so. So I know the homologue is called the AMY1A. And the normal function is to degrade the lipids in the gut, so will be secreted by the exocrine pancreas, I guess.
But it degrades sugars.
Yes, degrades sugars. So this is basically, we're looking at an endocrine function for something that normally is not really expressed in the muscle. Well, it gets re-expressed with aging or with stress, but otherwise has no strong expression or identified function in the muscle, like it is instead well known for the gut.
Thank you.
Thank you very much. I have so some of the questions you addressed a little bit. So one of it is, the last thing that you mentioned, that the sedentary muscle can just not doing nothing, but also secreting like detrimental myokines. Could you give us like a brief summary of which myokines or what is being secreted when we are sitting down like now?
So we didn't really do studies on this, but what we did was to profile different types of skeletal muscle, in this case from mice, with different types of wasting, so induced by cancer, by aging, and by a drug called dexamethasone. And so there are many secretive factors. Actually, the category of secretive factors is one of the top categories of regulated genes. So there are many that are modulated. We don't know which ones are just bystanders, so they are regulated, but they have no functional outcome on other tissues in the brain. But what I would like to speculate is that some of these, they mediate the detrimental effects of sedentary life on the brain as well as on other tissues, but we didn't do any functional studies on and the list of myokines, they are is a Cell Reports from 2021 that we published. Ant is the first author, and again is the comparison both by RNA-seq and mass spec of the responses that are induced by different types of wasting. So I can also send you the paper if you have interest in that, but unfortunately we don't have any functional studies at this point. And the flies, they could be used potentially for this type of studies, but it's less easy than in mice, where you can induce wasting in a controlled manner. In flies, these type of studies are a bit more complex.
Thank you. And also related to Fernando's question regarding the level of maltose with aging, what about the transporter? Does its expression also changes with age? Could it be like the brain is having less expression of the
So yeah, that's another great question. So these I don't know actually. But what I know is that is highly expressed in the brain compared to other tissues. And in humans, there are 4 of them. They are called SLC45, A1, 2, and 3, and 4. In flies is only 2, but what I know is that they are more highly expressed in the brain versus other tissues. I don't know though whether they are regulated by aging, but it's an important point. And I know about these NIPs associated with some cases of Huntington disease, but also in that case, I don't know if it is a loss of function or any other effect.
Okay. And thinking about the muscle, you mentioned like you came to this idea of checking, like to stress the proteasome because it will be a response of stress, mechanical stress in particular. So I have 2 questions related, one a little bit more serious. So one is like, is it known or has someone measured whether just exercise induces proteasome stress?
Yeah, so proteasome stress, I should say, is poorly studied compared to other types of stressors. So the type of stressors that were shown to be activated by exercise are the unfolded protein response, which is a response that is generally activated by unfolded proteins. And the mitochondrial unfolded protein response was also studied and shown to be induced by exercise. The proteasome stress per se has not been investigated in skeletal muscle, but we use it as a way to increase the number of misfolded proteins, because by partially inhibiting the proteasome, you basically induce a buildup in those proteins that are misfolded and that normally would be degraded by the proteasome. And so you could consider it another way to induce the unfolded protein response.
Yeah, well, I was thinking about if it were like directly proteasome stress, it will have an impact in UPR, as you say, but it could also have an impact in the level of expression of other proteins whose abundance, whose life, whose stability is regulated, that could influence also in cell biology. So it could be like more general than just.
So you're right. The proteasome not only degrades misfolded proteins, but is also involved in the normal turnover of protein. I should say that we also saw the same response with the VCP RNAi, which is involved in the degradation of misfolded protein from the ER as well as from the sarcomere in muscle. And so that is maybe more geared towards, again, inducing a perturbation of misfolded proteins only.
Okay, thank you. And the last question that perhaps is not serious, but thinking of the mechanical stress as a inducer of the UPR, will then a good massage be enough to induce amiral expression?
Sorry, I didn't understand.
A massage, a muscle massage?
Oh, I see.
So like the mechanical stress induced by, like physically massaging the muscle.
Yeah, this is an interesting point. I have actually heard of maybe a machine that will do that is proposed to be improving muscle function in old age, but we didn't do any studies. We cannot do it easily in flies. But maybe mice is possible. I'm not sure, but that will be actually an easy intervention. That's a great idea, actually. For the people that don't want to exercise, maybe.
Yeah, it's better to have a massage. Jogging early in the morning. Just kidding. Thank you. Is there any other question? Someone that would like to say something else? Well, thank you very much. It was really, really.
Thank you so much.
You talk. So, thanks.
Thank you.
Bye.
Bye.
Thanks.
Bye.
Bye-bye.
Gracias, adiós.